Hello, so the genes don't really work alone. Sometimes they need each other to create a trait, and sometimes one gene can block another one. To explain how such an interactions work, let's explain it with the four penguins' four colors, and in the end, you will understand why some traits need multiple genes to appear and why some genes can silence others completely. Well, let's start with a white penguin named Jessica and the white penguin named Martin. They loved each other very much and got married and had lots of babies, and all of them were purple. And from the last videos, you can see that there is no way we could ever gotten the purple color. And when they interbred with each other, we got both purple and white penguins. Well, how did the purple color emerge? Why did we get the 9 to 7 phenotypic ratio? To explain this, we have to dive deep. So we start defining genes A and D. A and D are converting uncolored compounds into the other compounds. So let's give us the synthesizing pathway. So colorless compound 1 turns into the colorless compound number 2, which turns into a purple pigment. So let's define the gene A, which converts the first colorless compound by creating some kind of the enzyme, which converts it to the second compound. Now, for the for the D gene, as you can see, it creates a different kind of protein which catalyzes the reaction which converts colorless compound number 2 to the purple pigment. Well, let's discuss two cases and let's start with the first one when both genes are mutant. So if both genes are mutant, they will create the deficient or mutated proteins which cannot do its activity. As such, it cannot catalyze this reaction step. As such, colorless compound gets accumulated, and purple pigments will not get created. Now, for the second case, when the D gene is not active, it creates a defective proteins which cannot catalyze the second reaction. Thus, the second colorless compound gets accumulated, and purple pigment cannot get created. As you can see, both genes are needed to create a phenotype, and this is defined by the complementary, which is the gene action when two or more genes interact with each other to produce a trait. Now, what phenotypic ratios does complementary produce? Well, we can just use the Punnett square to check it out. Now, for the first cross, when Alice and Martin got married, Jessica's genotype would be capital A, small d, while the reversed one would be the Martin's genotype. As such, we would get these gametes, and we would only have the heterozygous individuals. As such, all of them would be purple. All of their sons or daughters would be purple. Now for the second cross, when they interbred with each other, it is just the standard Punnett case for the heterozygous cross. And here are the genotypes, but we have to check each phenotype. So as you can see, for purple color to be created, we need at least one wild type E and one wild type D genes. And if we just check each square, we will get 9 to 7 ratio. So out of 16 penguins, 9 will be the purple one and the 7 will be the white one. Well, we just ended the complementary, but we got a new news from the far side of the penguin land where two brown penguins named John and Anna got married and had many and catastrophically many penguins. But they were black, brown, and also white. Well, why did we get three different colors? Do we have three different alleles, or why do we even got 9 to 3 to 4 ratio? How can you explain such a result? We have to investigate with same principles we used for complementarism. Well, let's define our synthesizing pathway once again, and let's also define the genes. So we have two genes. So gene C. So gene C determines if the pigment gets synthesized itself. For example, if both C genes are mutant, the organism might have a deficient melanocytes, and melanocytes create a pigment necessary for fur coloration. Now the gene A is giving pigment its color, so gene A gene A in its wild form might give the pigment brown color, and in recessive form it can give a black color. So let's start with the following. So first we take a C gene, which creates a certain proteins which actually create a pigment. They create a pigment. Now we have to now for the first case of A, as you can see, it synthesizes a protein which catalyzes the reaction to create a brown pigments, and for the recessive A allele, it just catalyzes the reaction which gives us the black pigments. Now we just need to check out the one case where both C genes are mutant, and as you can see, when they are both mutant, we will get some just mutated proteins which cannot do its function very well. As such, no pigments will get synthesized, and we will get defective melanocytes. As such, we saw that C gene actually controls if A gene gets expressed or not. It actually masks its expression. So this is called epistasis, and this is the gene action when one gene masks or controls phenotypic expression of another. In the first case, we saw that two genes work together to create a phenotype, but for this case, they both do that same, but one determines if other gets expressed at all, if its activity gets expressed at all. So, what phenotypic ratios does epistasis produce? Well, we just take a simple Punnett square, put all of the values, and we need to check. So if we have both dominant C and dominant A, we'll get the brown color. If we have at least one dominant C, we'll get a black color, and if we have the recessive C, we get the white color. And as you can see, we get 9:3 to 4 ratio. Well, in conclusion, complementary is a gene action when two or more genes interact with each other to produce a trait, and the epistasis is a gene action when one gene masks or controls phenotypic expression of another gene. Thanks for watching, and see you next time.